Thank you. So now back to the science. It's a pleasure to introduce Andrew Travers from University of Cambridge. Thanks very much.
Thank you very much, Dorothy, and thank you to you and the other organisers for inviting me here. Now, what I will do is to present a rather DNA-centric view of the world and follow up on what Charles and Craig have been talking about already. Now, this is just a very good place to discuss science, but what I'm really going to talk about is how the structure of DNA can affect the genetic organization of prokaryotic and eukaryotic chromosomes. Now, as you know, when you look at DNA, the original description of it was as a double helix. There's no reason why a double helix itself should be genetically important, but it confers one important property, which is, of course, topology. The other important properties of DNA that we should be aware of are first that it's informational. It has to code for proteins, RNA secondary structure, protein binding sites, and so on. Also, there are very important mechanical properties of the DNA which determine how it can be wrapped around, say, in nucleosomes, determine how easy it is to write in during supercoiling, and so on. All these properties, the topology, informational properties, and mechanical properties, are essentially interconnected. And if you want to understand how the genome works, you want to understand the connections between these different properties. Now, we know that if you're looking at the ability of DNA to bend, then the one important characteristic is the base pairs. Both information and mechanical properties are related to a first approximation to base composition. And what we showed about 8 years ago is that for a given DNA sequence, then the stiffness of DNA, as determined by AFM, correlates with the number of hydrogen bonds per base pair. So on average, if you're GC rich, you're a bit stiffer. Of course, the exception to this, as always, is poly DA DT, the homopolymer. But then you can say that each base pair is involved in actually about 3 hydrogen bonds. And so it's, if you like, it's an honorary GC base pair, and that maintains the rigidity of the AT base pair in GC. So it retains the rigidity of the AT base pair in homopolymers. Now, the other question about the mechanical properties is to what extent the individual base steps, and hence the DNA chain, are flexible. How much variation in structure can we observe? Now, Wilma and her colleagues several years ago had analyzed the variation in conformation of individual base steps in crystal structures of DNA alone and DNA in protein-DNA complexes. And what they found is, if I can get this to work, yes, what they found is that it's sequence dependent, it's also base composition dependent. So the inner circle here, was taken from one of Wilma's papers, is the free DNA. The outer circle is when you bind the DNA to protein, you get an extension of the envelope. And what you see here, protein TA, AT, they're both have a reasonable range in free DNA, and TA has a much larger range in protein-bound DNA. And if you look at GC, there's very little extension of the deformation in protein-bound DNA. So by and large, if you have, depending on sequence, the purine-pyrimidine step, you have more deformability. If you have the purine-pyrimidine step, you have less deformability, and the purine-pyrimidine are intermediate. And we can quantify these in terms of the melting energy of an individual base step or the stacking energy using values taken from work by Frank Kamieniecki. And what we've tried to do is to look at how the stacking energy and or the melting energy are distributed in bacterial chromosomes and eukaryotic chromosomes, in particular yeast chromosomes. And does this correlate with genetic function? And what we essentially do then is to take all the base steps, average them across a certain window, and then get a particular value, and then see how to take it from there. Now, what I'd like to start with is work we've done with some colleagues in Bremen, in particular Georgi Muskovishvili, about this distribution in coli. What I want to take first is a 300 chromosome experiment. And what you do is to take 300 genomes of the bacteria in a particular group, the gamma proteobacteria. You then, for each one, determine the melting energy across a particular window. This is a broad brush approach. These genomes will be different in length, so normalized for length, and then you average the lot. And what you see, if you have a large window, in this case half a megabase, is that the pattern of the melting energy is essentially symmetric about an axis from the top to the bottom. And that axis goes from the origin of replication to the terminus of replication. In particular, what you see, there's a gradient, more or less, of stacking energy, of melting energy from the origin round to the terminus for both replicors. Now, what this does is to try to filter out genetic noise. So you're looking perhaps at a fundamental bacterial chromosome, which is stripped of all its sort of bells and whistles. If you do it on a smaller window, you see a bit around the origin where the melting energy is less, which is good because that's where you want to melt the DNA. And the terminus, it's still very strong. And the question is, why do you get this distribution? And one possibility arises from a classic paper by Leroy Liu and Jim Wong, which has been alluded to already this morning, where if you have a DNA translocase, in this case, which is fixed and translocating along a fixed domain of DNA, so fixed at the ends, then in front of the translocating enzyme, you generate positive supercoils, and behind, you generate negative supercoils. So in the picture which we had here, then during the DNA replication, since this is obviously related to DNA replication, the regions of high melting energy are where the DNA will be more negatively supercoiled from replication, and the terminus is the region to be more positively supercoiled. And the question, is this relevant? Does it relate to the function? Now, this is, of course, an average of many genomes, and each individual genome is different. So if you look at the coli genome, what you see is a rather similar pattern, except that the gradient is more broken up and is different for both replicors. But essentially, the average melting energy is still higher near the origin, and particularly is the lowest near the terminus. These are the two. And if you go down to a much smaller window, then it breaks up even more, as you would expect. Okay, so can we say, is this distribution of melting energy correlated with function? And if we just look here, we have the melting energy in the outer ring. This is of coli. Then we look at gyrase binding sites, and you find the gyrase binding sites here and here are correlated with the regions of the highest melting energy. If you now look at the transcription units, which are sensitive to the the transcription factor sigma d, that's responsible for operates during vegetative growth, you see a rather similar pattern. That again, the sigma d promoters are they have the highest density in the regions of the highest melting energy. In contrast, the sigma s promoters, those which are expressed during stress conditions, they are located in the region of the lowest melting energy here. So there is an apparent correlation with function. Now, we published a preliminary report of this about earlier this year, just giving the phenomenology. And the important paper is one from Bordes in 2004, where he followed the superhelical density on a protoplasmid with the growth cycle of E. coli. And what he saw was that as the growth rate began to slow down, but not when it, that's before it's fully gone in stationary phase, the superhelical density of the reporter fell dramatically. And what we proposed was that what the input for this is you need oxygen and you need a high nutrient availability, and this gives you high negative superhelicity. And this is what Charles was talking about earlier. What we would then say is the gene expression pattern is coupled with negative superhelical density, and the chromosome superstructure is also regulated by the superhelicity, and that generates the temporal expression pattern during the growth cycle. Now, how do we want to look at this? Well, what we really want to know is how the genes are expressed during the growth cycle on a global scale. And what we do, first of all, is to take wild-type coli and mutants with mutants in the FIS protein or the HNS protein. So we're comparing wild-type with coli that lacks FIS or coli that lacks HNS. And we follow then the growth cycles, which is, come here, the one that goes up. And we follow the oxygen availability, the oxygen partial pressure, and you can see how that varies with the growth cycle. You can see here, as the cells get denser, there's less oxygen available. Till here, although the cells are still growing, the oxygen availability is very low. And then as you go into stationary phase, the oxygen availability increases because presumably they run out of other nutrients, and so they're not metabolizing so rapidly. So what you do then is do deep sequencing and follow the pattern of expression of these genes, of about 4,000 genes during the growth cycle, and see how it fits the chromosomal pattern. And what you see, and I hope you see, if you look at the wild type, here's the growth cycle. It goes through. This is the FIS wheel, this is the HNS wheel, and if we're going to start from 0, start now, you see immediately the genes around the top come out. Then there's a lag while the bacterium thinks or adapts, if you like. Then the expression is here. Then you go through stationary phase, and it's not just stationary phase. There are lots of different complex adaptations through that phase, which we don't know much about. If you look at the FIS and HNS wheels, you'll see that the pattern of relative gene expression throughout the chromosome is different. And they're widespread changes. These are largely quantitative differences and some qualitative differences. So we would say here, let's see here, it says just shut. At the end, it just shuts down completely. So this says that the genes closest to the origin are expressed very highly during the initial growth rate, the initial phase of rapid growth. They're then shut off, and they're then shut off, and we can quantitate this by looking at genes which are sensitive to raised superhelical density, or genes which are switched on when the superhelical density is lowered. We call these genes HIP and REL respectively, and what you see in these wheels is basically the time course from 0 time at the center moving out through the growth cycle to final stationary phase on the outside. So if you look during the initial phase of exponential growth, you see that the genes that are expressed at high levels are largely those that are stimulated by high superhelical density. Then when you shift out into oxygen deprivation, these are switched off. The pattern with the rel genes is not the converse of that, but by and large, you see that the rel genes are not greatly expressed during the early phase, apart from this cluster. But when you move out, then more gets switched on. So there is an approximately reciprocal arrangement, but it is not perfect. And this upper wheel just shows you the density of transcription. And again, you see that the density of transcription is localized to the oriproximal region during the initial phase of exponential growth. And when you switch to the oxygen-deprived region, you get it transfers to the other end of the chromosome. This, in the same system, you can also show that during the initial phase of the exponential growth, that transcription from the lagging strand of the replication is favored, or leading strand, sorry. Now, what Charles told you about the naps, he gave an excellent introduction, was just one aspect that I would like to emphasize, and that is that naps constrain DNA superlicity, and they constrain it in different ways. The FIS protein, for example, can constrain a plectineme with a fairly loose plectineme, or its particular regulatory sites. It can constrain a toroidal DNA, wrapped DNA, just like half a nucleosome. The HNS and its paralog STPA can constrain a rather tight plectineme. These are all negatives, these superquad. HNS, HU can constrain a toroidal form. Not all naps constrain negative superlicity. The DPS doesn't. Now, so we can ask, if the superlicity varies during the growth cycle, to what extent does NAP synthesis, this is not NAP total concentration, which Charles was talking about, to what extent does does NAP synthesis vary? So this is a very busy slide, but as a region where there's high negative superlicity, low negative superlicity, and the NAPs which are synthesized during high negative superlicity are two. There's FIS, and there's STPA, the HNS paralog. HU and HU alpha is reasonably synthesized, HU beta at much lower level. Then if you move to the region of low superlicity, you get HNS maximal, you get low maximal, you get IHF beta maximal, and you get HUP-A and HUP-B the maximal. These are the HU alpha and HU beta. The ones that come in later are IHF alpha and the DPS protein. This correlates with the transcription factors that RPOD, the vegetative transcription factor for RNA polymerase, peaks around this region here, whereas RPOS peaks around here for where your superlicity is lower. And this correlates beautifully with the experimental observation from Ishihama that RPOS containing RNA polymerase prefers to transcribe DNA of a lower superhelical density than RPOD containing RNA polymerase. So if I just summarize sort of what I've said, what we suggest is that you start off with a fairly compact chromosome when you immediately prior to shift up. You add your oxygen, nutrients, and then, as Charles said, the ATP-ADP ratio increases, dries activity is up. And this then leads to a change in the conformation of the chromosome. And if you like, it's an opening up. And this is an increase in negative superlicity that opens up the chromosome. What you will have, as Nancy beautifully showed, is that there are these are plectinemes of different senses. You have right-handed ones or left-handed ones, and you might even have open ones. And this is simply what you would expect if the chromosome was dynamic and there was twist-derived partitioning dependent on transcription or whatever. You then get DNA replication, and that will then give you positive or superhelicity towards the terminus and negative towards the origin. And both this initial winding up and the DNA replication will maintain a gradient of superhelicity from the origin to the terminus. When you have oxygen depletion, you then compact the chromosome. Those genes that are stimulated by high negative superhelicity turn off, and the rel genes turn on. So that's the model, and it's a very broad brush. I'd like to move on now to eukaryotes, in particular to yeast. And this is work done largely with Boris Adrian at the Department of Genetics, Cédric Vedant and Alain Aniodo at the Laboratoire de Physique in École Normale de Lyon. David Clark and Hope Cole provided some of the data. My nephew, Alex Window, talked to me about information, and it's all been done with many conversations with Michael Thompson, who's sitting in the audience. Okay, and what we do is to do this now on a much more detailed scale than we did for coli, where we look at the average stacking energy here, average melting energy on a 50 base pair running window across the yeast genome. We correlate this with nucleosome positions in vivo, nucleosome positions in vitro, and the genetic map. We also we look at the curvature of the DNA, the bend, if you like, the preference to bend in a particular direction. We have all this on the computer, and just to cut a long story short, if we do this, then we can ask how is the melting energy or stacking energy distributed in terms of the melting energy across the whole chromosome. And you can see this is kilocalories per mole per base step over a range for the yeast, and there's a peak here which corresponds to the base composition of yeast. It trails off on both sides. There are various outliers, and these outliers are important. Then you can ask, how is this distribution reflected in the genetic organization? If you look at the pale purple, this is genes, and genes are most abundantly represented just to slightly skewed to the GC-rich side. The 5' regions, the 5' UTRs, are fairly well represented here on the far GC-rich regions. They're also pretty well represented here to the other on the 80-rich side of the energy peak. But the most striking thing is this patch here, 64%, 48%, 22%. There's a strong skew of the 3' UTRs to the region with low melting free energy. Now this is interesting, and if you show you one example, you can see there is a particular one here where the melting free energy is low, and this is polished on a minus delta G scale, so if a peak means it's low, and this corresponds to a region of low nucleosome occupancy in vivo, in vitro. Genetically, it is at a region where there is a potentially convergent transcription of two genes. And so it's exactly like the terminus in the coli chromosome, where you have convergent replication of two ribosomes. Now, if we now look at the sequence, and you see it is pretty AT rich, and in fact, it's a very nice T88 run, and many of these 3' UTRs have this characteristic. They're basically very rich in purine pyrimidine dimers, purine pyrimidine base steps, and that's why they have this low stacking energy. So what we suggest then is essentially that we have a region where you have convergent transcription, or you don't even have to have that. You have in the TATA you have a topologically sensitive region. As you generate positive superhelicity downstream of the transcribing polymerase, that will lead to distortion here in exactly the same way you get the transmission that Craig was talking about. This is TA. And one characteristic of TA, of course, is that it is the most highly preferred binding site for topoisomerase 2. So it's a good way of taking out these supercars. That will also prevent that if you only have one gene being transcribed, prevent any superlicity disrupting the chromatin structure of a neighboring gene. And you also know that since topo2 can co-purify the chromatin matrix, you could have a point of attachment there, which would then essentially, so that this region would essentially constitute a chromatin barrier. It could be dynamic, it could be static, we don't know. Okay, so that is the same Lou-Wong principle. Now we can move on to nucleosomes, and I'll try and be very quick on this. If we look at nucleosomes, this is a plot that Cedric Vaillant did, just looking at a particular region of the yeast genome. And what you shows is nucleosome occupancy in vitro, this time, in red, and stacking energy in blue. And you can see there is a pretty good correlation between stacking energy and nucleosome occupancy. I should say that Cedric put the stacking energy plot the other way up. So in fact, this bit down here means that the stacking energy is very low. So basically what we're saying is that high nucleosome occupancy in vitro correlates with high stacking energy. Intuitively, that's not quite what you'd expect, because you expect if you want to wrap the DNA around the histone octamer, you want it to be flexible, not stiff. But the other important thing is if you look at the open reading frames, they too correlate with regions of higher stacking energy and high nucleosome occupancy. So this is good, but what does it mean? Now I just want to sidetrack a moment and look at some in vitro data. And the in vitro data, what we do is to take two sequences of DNA and we reconstitute nucleosomes from them in vitro in a competition situation. And then we can vary the conditions so we can see how the competition varies when you alter the conditions. And what we take first is the 601.2 nucleosome, which is a nucleosome sequence which is selected on the basis of its very high affinity and the selection conditions. And we compare it to a nucleosome here, which is from a yeast gene and corresponds to a known nucleosome position. So this is very AT rich, this is less AT rich. And you can see under the conditions where this sequence was selected, it really wins very well. But the K1 over K2 is high. That's fine. And this just on the abscissa we have the average stacking energy. So this has a higher average stacking energy than this one. Now then, if we do the same thing, where this reconstitution is done under high sort conditions, and you gradually lower the sort, so you then do the reconstitution under low sort conditions, you reverse the sequence preference. And in fact, the difference between the low sort and the high sort conditions is about 300 to 400 fold, which is fairly substantial. If you raise the temperature slightly, and here we have a range of sequences, then, but still at low sort, you still get the sequences with the highest stacking energy having the lowest affinity. So you can get a complete reversal. You can also get some reversal by raising the optimal concentration, and the effect is minimized by the by temperature. But the important thing is you can see there might be here, when we're looking for it, a region where the average stacking energy has no effect on relative affinity. Now, how do you think about it? The way we think about it is that formally nucleosome formation is a two-step process. You get trapping, and wrapping. Trapping involves the binding of the DNA and then the bending around a little bit of the histone optima. This process will obviously be facilitated if there's already a bend of the correct orientation of configuration in the DNA. Then the wrapping will follow. Once you've trapped, you can wrap. K1 over K2 is the initial binding. This is the important factor. When you reconstitute a high sort, K1 is very similar to K2. They're both very rapid. At low sort, K2 is minimized. And so what we would argue is as you lower the sort, then the on initial binding, the DNA can still wriggle. And as long as the rate of wriggling is greater than the off rate, you enhance the probability of trapping. So this is essentially can be dependent on entropy. This is the deformation is an enthalpic penalty. And this is going to be sequence dependent because you can just think of DNA in terms of configurational space. If you are highly flexible and isotropic bending, you get lots and lots of different configurations. If you're stiffer and the bending is in a preferred direction, which is sequence dependent, then you get a much more constrained range of configurations, and you increase traffic if you're in the right conformation. And you can look at this in terms of what we call the thermodynamic cycle. You've got to always associate the optima, free optima, and DNA, and then in vivo you have the chromatin remodelers, and for chromatin remodelers, of course, it's very light because the nucleosome the optima is already there. K1 will be very much greater than K2, and there you will lose your sequence preference, and then you can move the optima to any position, as has been shown. And then finally you get dissociation. So the question then is what happens in vivo and what happens in vitro? And what we've done is to look at the dependence of nucleosome occupancy on the stacking and melting energies, and in vitro and in vivo. And here this is the in vitro result. As expected, what we find is that as you increase the stacking or melting energy, then occupancy increases, and it's still going up. These are the ranges where it's statistically valid. So that's in vitro data, and there we say that this is essentially driven by minimizing the entropic penalty. If you then look in vivo sequences, what you see is the same thing. This is in yeast, that there is a strong dependence of nucleosome occupancy on the stacking and melting energies. Again, the stiffer the DNA, the greater the occupancy, and in principle, the greater the affinity. Now, this is for yeast, and it's not true of all organisms. If you look at Drosophila in tissue culture cells, you can ask take all nucleosomes, what is the fit to the best bending sequence, which is a sequence for like these 601 nucleosome I just talked about, and how does it correlate? And the answer is it doesn't. There's no correlation. This is now free DNA, and again, there's no correlation. If, however, you knock out the remodelers, you begin to see correlations. If these are cohesins, if you knock those out, no effect. So basically, the default state is binding to a sequence that is anisotropically bent, and that the remodelers then can remove it from the default state. Now, it's also true that this varies during development. So that if you look at early Drosophila embryos, very early, there's a very good correlation. As they develop through to larvae, this correlation decreases. So basically, is there a preferred position for sequence for nucleosome positioning and so on? The answer is, being biology, as always, it depends on the system you're looking at. So, now, how to explain this? This raises a number of questions. And the questions are these. So why does high nucleosome oxygen increase with high stacking energy? And what we found before is that regions of high bending sequence periodicity in yeast are associated with the more AT-rich sequences, but these AT-rich sequences actually have low occupancy. This is a paradox. There's how can you code for both positioning and protein information? And I think I've talked about DAT already. And so the answer, I think, is that what you want to look at then is how in terms of the stacking energy and the available information, is that if you go to very high GC, then your range of possible configurations, what we call the Boltzmann microstates, is much lower, and this then increases with AT content. So once you get to 100% AT, you can either have poly-DAT up here or poly-DADT, and you have a huge range of possible configurations. So this region, but if you have a sequence moderately DAT rich, like here, then you're sort of on the limit. And the reason you're on the limit is that the minimum stacking energy, or the minimum number of these states you can have, is increases with AT content up to a certain point. So this region here is a nucleosome-friendly zone. It's both stiffer, and as you go down, then you actually require less information content to fix the nucleosome. And the reason you require less information content is actually because it's stiffer, and you have a greater probability of then incorporating an appropriate configuration for binding. So just to summarize, this is the data from yeast. You have this range of melting energies. These are promoter nucleosomes here on the high AT-rich side. This is a very GC-rich nucleosome here. The UTRs are out here, and these are the peaks for the different genetic elements: the 5' UTR, the 3' UTR, and the genes themselves. Okay, and so if I can just summarize finally and take all the people involved, this is how you see a gene. These are your standard GC-rich nucleosomes. They don't like it here at all. This nucleosome has a lower affinity and can easily be removed when you activate the gene, and so on. Okay, thank you very much.
We have time for one question while Lynn's setting up.
Steve.
So you raised the issue of topologically closed domains being formed by the binding of some of the nucleoid proteins.
Yes.
And that raises a certain issue that maybe could be addressed to all three of you who've spoken this morning. And that is, when you constrain small domains, say a few hundred base pairs of DNA,
Yes.
you potentially change the balance of twist and ride in terms of
Absolutely, yes.
Yeah. But I've not seen anywhere in the literature any indication of this. And I'm wondering whether maybe there's something I've missed. Because you would expect, if you have a small topologically isolated domain,
Yes.
that you ought to get transitions that involve changes in DNA twist occurring much earlier in terms of supercoiling than would be the case if they were in, for example, a bacterial chromosome that's not constrained.
I agree. This is a very important issue. And the way I look at it is if you have a protein which untwists the DNA at exactly where it binds, so that's fully constrained, then that untwisting in a closed domain will be reflected in changes in rise outside. And one possible example of this are the H and GP proteins, which bind to nucleosomes. Okay. They melt the DNA. They bind. They will generate positive superhelicity immediately outside, and the suggestion is that that positive superhelicity will then counteract the negative superhelicity of the wrapping, negative rise of the wrapping, and assist the unwrapping. Now, experimentally, we know that H and GB proteins facilitate the unwrapping of nucleosomes. Whether this is the mechanism, we don't know, but it's the speculation.
That's interesting. Thank you.
All right, let's thank Andrew again for this.